Okay, so in this video we're going to talk about the anatomy of the kidney as well as the anatomy and a little bit of physiology of the nephron, which is the functional unit of the kidney. So right here we have a cross section of a kidney if we cut it cross sectionally. And the first thing you need to know about the kidney is how it's shaped. So it's kind of this like bean-like shape, right? Like this with this little indent here. This indent actually has a name, and it's called the hilum. So I'm not labeling any kind of structure here, it's just this indent. It's just a little tricky to label on the diagram. So that's the hilum, and the hilum is the point of insertion for blood vessels, nerves, as well as the ureter, which is actually this tube right here. Alrighty, so I guess the next thing we should talk about is the outer lining of the kidney, the little membrane surrounding it. That's called the renal capsule. Alrighty, and then the next thing is that the kidney has two different layers, you could call them. So the first is this layer all in these blue dots. That's known as the cortex. It's also called the granular portion of the kidney, which is why I drew it with all these little dots like that, which refers to when you look at its texture, it is granular, and especially if you look at it under a microscope, it does look very granular like that. So that's the granular portion. And you'll see it's like this whole like kind of outer layer, but then it also dips in between these spaces between these red shapes that we'll talk about in a minute. These are known as renal columns, these spaces here that extend down. Let's see. Alrighty. So that covers the cortex and the medulla. The renal papilla comes in contact with these little divots right here that we'll talk about in a minute. These are called the calyces is the word for the plural. So each one of them is known as a calyx. And there are these little indentations in this space, which is known as the sinus. So the sinus here is a cavity which is connected to the ureter, and in between the sinus and the ureter is this space here, kind of a funneling down into the ureter. That's known as the renal pelvis. So when the kidney produces urine, this is where it comes down through this space into the sinus, and then goes through the renal pelvis into the ureter. Through the ureter, it would go into your bladder, which could be excreted out of your body through your urethra. And that's how the urine is excreted out of your body as waste. So these are basically all the little structures you need to know. The last thing I want to talk about is, I'm going to draw the nephron more in depth in a minute, but what you really need to know here is that there are certain parts of the nephron that are in the cortex and certain parts that are in the medulla. So if I just drew like a really elementary drawing of the nephron, here's the renal corpuscle, which we'll talk about. you have convoluted tubules, and then you have the loop of Henle, and then you have another convoluted tubule, and it empties into a collecting duct. So what you need to notice here is that the loop of Henle is inside the medulla, whereas most of the rest in the neuron is actually in the cortex. And not all neurons are the same. Some are, you know, just barely in it, like this much in it. Some are really deep in there. It all depends, but that's the different portions in each section. And I'm going to go into the anatomy of the nephron in just a minute, but I just wanted to, you know, make sure you understand that before I talk about the nephron anatomy. So I hope this helps you understand the kidney. It's pretty basic, you know, just straight anatomy, basically, not much physiology. Okay, now let's take a look at the actual nephron, which could be described as the functional unit of a kidney, meaning it's the smallest piece that does the function of the larger organ. So to get started, let's talk about the renal corpuscle, which is composed of two structures. So first we have the glomerulus, and then next we have Bowman's capsule. And then together they're called the renal corpuscle, like I said, so I'm just going to write that above both of them. Okay, so those two structures consist of comprise the renal corpuscle. The glomerulus is actually a large, well, I guess not large, it's a tightly knitted bed of capillaries. It kind of forms like a little ball, and then sort of wrapping around it but not totally engulfing it is the Bowman's capsule, which is actually the end of a hollow tube that's been modified, so it kind of fits around this capillary bed. They're extremely closely interacted, and we can talk about a little bit more specific about how those two are connected and stuff in a minute, but first I want to go through the rest of the anatomy real briefly. So that's the renal corpuscle, and the glomerulus actually is a capillary bed coming off of the afferent arteriole, which is right here. Which would have come off of the larger renal vein. So we go from the afferent arteriole to the glomerulus, and then that associates with the Bowman's capsule. Like I said, the Bowman's capsule is part of a hollow tube, and the whole rest of this nephron right here is all one tube, but I've labeled them according to color differently because they have different names, they have different functions, different structures. So it's actually one full piece, but they're a little different. So that's how we're going to talk about them. So extending off of Bowman's capsule is the proximal convoluted tubule. Which is the space right after the Bowman's capsule. And then after the proximal convoluted tubule, we have the loop of Henle, which is this entire structure in green here. Again, same. It's all one tube, but it's a different part. So that's the loop of Henle, and it's actually, you could break that up into two different parts. We have the descending limb, and we have the ascending. right, so that's the loop of Henle in green. And then following the loop of Henle, we have the distal convoluted tubule. So we have two convoluted tubules. One is the proximal convoluted tubule, one is the distal. So distal meaning farther away, and that would be from Bowman's capsule, where the proximal is very close, and that's why that's named that way. And then from the convoluted tubule, the whole entire nephron empties into the collecting duct. Should be here. All right, so that's the basics. Now let's go through and talk a little bit more detail. First, what we're going to do is talk about the remaining blood vessels. I'm going to have to move this. All right, so like we talked about, the glomerulus is a capillary bed, which means coming off of it there should be another blood vessel, and that right here we're going to draw, and that is known as the efferent arteriole. So we have the afferent arteriole, we also have the efferent arteriole. So this is the efferent arteriole, and this leads into a second bed of capillaries that surrounds these tubules here. So we'll just draw them wrapping all around. It's a big bag of the capillaries. Okay, and these capillaries are called the peritubular capillaries. Makes sense because they're surrounding the tubules. And then from the capillaries, we're going to have the renal vein coming off. So that's the entire system of blood vessels. So we go from the renal artery, which I haven't drawn here, to the afferent arteriole, into the glomerulus, which is the first capillary bed, out to the efferent arteriole, which leads to the second bed of capillaries here, the peritubular capillaries, and then we exit out the renal vein. And that would be the flow of blood through the nephron. I shouldn't say through the nephron, associated with the nephron. So first thing we want to talk about is because we have two capillary beds here, that means that this is a portal system. So you should be familiar with the idea of a portal system. A system just means that before returning back to the heart, we don't go through one capillary bed, we go through two. So this is how this works here. So this is a portal system. Next, we're going to talk about a few different modifications in the structures of the nephron and how that relates to the three functions of a nephron. So up here I'm going to write the three functions. So there's three functions, or you could think of them kind of as like three stages of the nephron. The first one is called filtration. Number two is reabsorption. And the third is secretion. So this is kind of how we're gonna keep these functions in mind as we go through and talk about the anatomy and the structures of the nephron. And remember, you always want to keep structure versus function in mind when you think about anatomy and physiology. So first things first is filtration. And filtration occurs here at the renal corpuscle, which is the first thing I pointed out. And the renal corpuscle is actually modified in several different ways to allow filtration to occur. So what happens is you have a certain amount of blood pressure in these blood vessels here in the capillary bed and the glomerulus. And what it does is it pushes against the walls of the capillaries, and it actually gets pushed into the Bowman's capsule. So the Bowman's capsule and the glomerulus are very closely associated, and they're both modified. So I'm going to draw here what the filtration barrier looks like real quick. So we have the basement membrane, which you know is an acellular component of a tissue between two different cell types. So we have the basement membrane. And the basement membrane is between the capillaries, draw them in red, the capillaries of the glomerulus, right here. These are the cells of the capillaries, the walls. And then we also have in the Bowman's capsules these things called podocytes, which pod, the root pod means foot, and they look like cells like this that actually have little feet. And these podocytes kind of wrap around the walls of the capillaries. Okay, so these spaces here, called filtration slits, is what allows the filtrate to actually, the components of the blood to flow from the glomerulus into the Bowman's capsule and create filtrate. So anything that is brought from the blood into the Bowman's capsule, those components, that liquid that is formed, is called filtrate. So how these filtration slips are formed are the capillaries in the glomerulus are actually fenestrated, and fenestra, the Latin word fenestra, actually means window. So these capillaries have tiny spaces between the cells which allow things to pass through them more easily. Also, the podocytes here kind of wrap around the capillaries and allow very close proximity. So the podocytes of the Bowman's capsule, along with the fenestrated capillaries of the glomerulus, create that filtration barrier that allows the filtrate to be formed, very efficiently. Furthermore, the basement membrane is negatively charged, which means that large proteins or large molecules like proteins cannot make it through because of their charge. So they're going to stay back, and like I said, only the things that aren't going to go into the tubules to form filtrate are molecules like proteins and cells. So large molecules such as proteins or cells do not go, and they do not produce filtrate. So the things that could produce filtrate are like water, ions, solutes, things like that. So once the filtrate is formed, it's going to come into the tubule of the Bowman's capsule, and then it's going to flow into the proximal convoluted tubule. So we already talked about the first function, which is filtration. Now we're going to talk about reabsorption. So the proximal convoluted tubule is structured in a multitude of ways that help reabsorption happen. First of all, the tissue type is simple cuboidal, which means that it's very thin. Things are able to pass over that barrier quickly. Furthermore, it has aquaporins in it, which can help bring water over the barrier easily. And they also have microvilli on the surface, which helps increase surface area for reabsorption. And lastly, they have a high number of mitochondria, which produces ATP needed for the active transport of solutes. So reabsorption means that we're going to take the components of the filtrate, both the solutes and the water, and we're going to push them from the tubule back into the blood through these peritubular capillaries. So we just went from blood vessels to the tubule. Now we're going from the tubule back into the blood vessels in the second capillary bed, the peritubular capillaries. So in the proximal convoluted tubule, I'm going to throw some numbers at you. I don't think they're terribly important to understand the structure, obviously, and stuff, but it's just a little bit of physiology for you. 65% of all the water and solutes that come in this initial filtrate are going to be reabsorbed in the proximal convoluted tubule, always. After the proximal convoluted tubule, where the filtrate enters the loop of Henle. The descending loop of Henle and the ascending loop of Henle are structured differently and therefore have different functions. So the descending loop of Henle actually reabsorbs water but not solutes, whereas the ascending loop reabsorbs solutes but not water. So first let's talk about the descending loop. The descending loop is made of simple squamous epithelium, which again is very thin for a reabsorption barrier, and the descending loop of Henle also has many aquaporins, which help it reabsorb the water, like I said. So 15%, another 15% of the water left in the filtrate is going to be reabsorbed. So we lost 65% of the original filtrate water to the proximal convoluted tubule, and then another 15% here. So at this point, we're going to lose 80% of the total volume of water from this initial filtrate formed at the renal corpuscle. In the ascending loop of Henle, we do not have any aquaporins, which means no water can be reabsorbed. And the other difference is that the filtration, or the tissue, is a little bit thicker. It's actually cuboidal or columnar epithelium, so it's a little bit thicker on the side than the simple squamous. And here we're going to transport the solutes back into the blood. So we're going to reabsorb solutes, and that number for you is and that number for you is 25%. So 25% of the original solutes in the filtrate are reabsorbed at this stage. So again, we lost 65% of the solutes here, and then 25% here were reabsorbed. So that means we've reabsorbed 90% of the solutes. So those numbers again are 80% of the water and 90% of the water. So once we get to the distal convoluted tubule, here in brown, we only have 20% of the original water from the filtrate that was formed in the renal corpuscle, and only 10% of the solutes. So reabsorption also occurs at the distal convoluted tubule, but it's a little more specific. So there's actually a specialized area here which I didn't point out earlier. So right here there's actually a specialized area where the afferent arteriole and the distal convoluted tubule are very close in proximity. Right here we have a region called the juxtaglomerular apparatus. So what this is basically is that both the distal convoluted tubule and the afferent arteriole are highly specialized in this one area, and it allows the nephron to basically be able to control the rate of its functions, more or less. That's, I guess, a simplified way of saying it. So the distal, let's talk about the arteriole first. The afferent arteriole has specialized cells called juxtaglomerular cells, which are actually mechanoreceptors. So they're able to sense stretch. So depending on the blood pressure in the artery, in the arteriole, they will feel that stress being put on the walls of the arteriole, and it can vasoconstrict or vasodilate in response to control the blood pressure in that space. Furthermore, the distal convoluted tubules specialized in this area of the tubule in the tissue is made of highly packed specialized columnar cells, which actually has chemoreceptors, which are able to measure the concentration of salt in the filtrate and then understand how much salt the body needs or doesn't need. So what I'll get to now is that these, and that area where there's highly packed columnar cells is called the macula densa. So reabsorption takes place in the proximal tubules and the loop of Henle, and it also takes place in the distal convoluted tubule. So, of the remaining salts, now we talked about solutes already, there could be a lot of different solutes, urea, potassium, vitamin C, something like that, but of course there's salt in the filtrate. So of the remaining salt left, 8% of the remaining salt is reabsorbed back into the blood no matter what. The other 2% that's remaining can either be excreted or reabsorbed based on the body's needs. So the chemoreceptors can sense the osmolarity and can adjust. So if it's excreted, it'll stay in the filtrate and then be excreted as urine, as waste, or it can be reabsorbed depending on what your body needs. So so that was salt. The distal convoluted tubule, just like the ascending loop of Henle, does not have aquaporins in it, which means it cannot reabsorb water. That being said, under special circumstances, say you're really dehydrated or something, your body needs to retain this water, it can't give up as much water as would normally happen. Like I said, there's 20% of the remaining water that was formed in the filtrate left. If your body needs that water, ADH, which is a hormone, ADH can cause the production and the insertion of aquaporins in the membrane of the distal convoluted tubule, and thus water can be reabsorbed back into the blood if the body needs it. If this happens, not all of the water can go back. Of the 20% remaining, 19.8% of that water maximum can go back. You still need some water left in the filtrate in order to dissolve those solutes to form urine. You can't take all the water away, or else the solutes would not be able to be dissolved. So that was reabsorption. So reabsorption takes place all over these tubules, and then filtration takes place in the renal corpuscle. What we haven't talked about yet is secretion. So if reabsorption is going from the tubule back into the blood for the body to use those components, secretion would be the opposite. So if there's something in your blood that the body doesn't need, let's say it's a waste product like urea, it could be brought back from the blood into the tubule to be excreted as waste. So because here in the proximal convoluted tubule, 65% of that filtrate gets pushed out, say that a lot of that was urea and we don't want that, we can secrete it back into the lumen of the tubule, or the opening of the tubule, and it could leave the body as waste. So that's how secretion works. Now we talked about the three functions: filtration, reabsorption, and secretion. Let's just go over it real quick. Filtration occurs at the renal corpuscle. That is where components of the blood other than proteins and cells are pushed into the tube known as the Bowman's capsule. From there, we have reabsorption, which occurs in the proximal and distal convoluted tubule, as well as the loop of Henle. And reabsorption is when you take components of the filtrate inside the tubule and bring it back into the blood for the body to use. Then we have secretion, which is the opposite of reabsorption, which is when we take things we don't need in the body from the blood and move it back into the lumen of the tubule to be excreted as waste. So after these three processes occur, everything at the end of the distal convoluted tubule will empty into the collecting duct. Now, once it gets to this phase, now it's urine. It's no longer filtrate, it's urine, and it can be excreted as waste. So several different nephrons can empty into the same collecting duct, and then those collecting ducts will empty into the renal pelvis inside the kidney, and then it will leave through the ureter and leave the body as waste. So that's how the nephron looks and how it functions. I hope that was helpful for you guys. I know it's a lot of specifics with the percentages and everything, but at least I could help you with the anatomy and the way it looks and everything. So I hope that helped.